All right. So, so when we talk about the properties, like when we understood like what enzymes are, or what is the chemical nature of the enzyme, and how enzymes behave, and how they decrease the energy level during the chemical reaction, we have studied. So for the enzyme to act at its optimum levels, we have to understand what are the properties that affect the enzymatic activity, right? So when we talk about what are the factors that will either increase or decrease the rate of reaction, okay, of an enzyme, that would be temperature, pH, and then we have substrate concentration. Okay, so these are the factors. So let's see how each of them affects the enzymatic activity. So first one, let's talk about temperature. So when we talk about temperature, temperature is very understandable factor. Why? Because if you observe any living system, for example human beings, which are warm-blooded organisms, we maintain a specific temperature for all these biochemical reactions to take place within our body. So why temperature becomes such an important factor? If we talk about an inorganic chemical reactions, as I told you, the rate of reaction increases for every 10-degree change. So usually for these reactions, higher temperature is equals to higher, higher the rate of reaction. But the enzymes do not behave the same way like that of your catalysts. So the difference between the enzyme and a catalyst is one of the major factors is temperature. How does it influence? If you see the composition of I mean, like what enzyme is made up of. Enzyme is nothing but a protein, right? So if you talk about the protein, protein is a structurally functional molecule. That means the structure of the protein is very important to determine its function. Understood? So now when we have the temperature, what happens? Higher the temperature we go. Now let's say that this is a chemical reaction or enzymatic reaction that has happened. Now we begin the enzymatic reaction and we start from at a certain temperature. As we increase the temperature, now what happens? Slowly the rate of reaction also increases. Up to what point? Up to a certain point. So at this point, you can see that there is a highest level of productivity. I mean, like there is a highest level of chemical reaction or enzymatic reaction that is happening. How do you know this? Because you're getting more products. Okay, so it goes in a very positive direction. So what happens next? Once you reach this point, it doesn't remain like that as you keep on increasing the temperature. So as you move on with the temperature, like as you increase the temperature, what happens is now the slowly the enzymatic activity decreases and finally it comes to an halt at a certain temperature. Why does this happen with respect to enzymes? First thing, since proteins are very sensitive to the rise in temperature, now what happens? Proteins are made up of peptide chains, and peptide chains are held by different types of bonds, such as ionic bonds, disulfide bridges, okay, or hydrogen bonds, okay. And sometimes what happens is there is a peptide bond that is between the amino acids also. So as the temperature increases, this sensitive structural configuration of the proteins with the rise in temperature, what happens? They change, they get damaged, or they get broken. So when this happens, now obviously the enzymes will lose its property. Why? You know how enzymes react. They go and recognize the substrate at a specific region, right? And that happens at the active site. So once the enzymatic structure is destroyed, now that it can no more go and attach to these active sites. So that is how the temperature influences. Now we'll take two examples. One example is when we raise the temperature, I told you that the protein degenerates and now it becomes dysfunctional and it can no more act as a normal enzyme. It doesn't it doesn't produce any results. Okay, it's denatured. Whereas what happens when when we talk about the lowering the temperature? As we lower the temperature, what happens? See, now the movement of the enzymes will, the kinetic energy of the enzymes will be lost and the enzyme structure will be intact. Okay, so now what happens? The enzymes literally freeze. Okay, and they do not I mean, like the activity of the enzyme will be same, but at the lower temperature, what happens? The activity completely stops but the structure remains the same. So this is what happens in the lower temperature. So one example that I think I already mentioned is the cryopreservation. When we freeze tissues or when we freeze some organisms, like we are trying to do that, okay, or microorganisms if you take, what happens is the cellular functions actually shut down at lower temperature. So because of several reasons. So that is why temperature, as we go down, the structure remains same and the function of the enzymes is shut down. So now as we increase the temperature, the enzyme can react back again and then perform the same activity like before. But in higher temperature, it is not possible. Okay. So next one. Another factor that affects is the pH. Okay, so we have two examples. For example, if you take thermophilic organisms, they are specialized enzymes. Okay, they are specialized enzymes. Basically, they are protected by some proteins. Some of the proteins, they help these enzymes survive, okay, in that extreme temperatures. So that is why they are able to produce enzymes that can sustain very high temperatures. It's very exceptional. It's not in all cases of organisms. Okay, only special organisms have that. Okay, now next one is the pH. Okay, let's talk about the pH. What happens with the pH? Now I already we discussed about the factor pH, how it affects the enzymatic reaction. Again, one more time to revise, what will happen with the pH? Enzyme is very specific to the pH change. One of the best examples we can take is our digestive system. So if we talk about our elementary canal, our stomach, right? In the stomach, our pH is around 2, right? The pH of the stomach is around 2. Whereas if you talk about the small intestine, it's around 7.8 pH. See, from almost very acidic to alkaline, the pH changes. Now, both your stomach and small intestine have very important functions to perform, and both of them are completely different. If you see in stomach, there are certain enzymes, okay, which can act at only acidic pH, okay. For example, pepsin. So the pepsin can only become active at the acidic pH. All right. Whereas in alkaline pH, there are so many enzymes, okay, that are produced in your small intestine that are active only in the alkaline gut environment. So pH also is very, very important. When we talk about both temperature and pH, at one particular pH or at one particular temperature, we will have the highest productivity. So this point where we get the highest rate of reaction or highest number of products for a chemical reaction. So this is called as optimum pH or optimum temperature. So let's see if we have that. Okay, you can see that it is actually optimum pH or optimum temperature. So anyways, it's fine. So another factor that affects the rate of reaction, especially in the enzymatic activity, is concentration of a substrate. Excuse me. All right. Okay, students, are you able to follow? Is it okay? Can you see the board properly? Are the letters visible? Let me know. Yes, perfect. So we can continue, right? Chalo. Sorry. Yeah. So this is a very, very important concept, how substrate influences the enzymatic activity. Now, what is a substrate? For every enzymatic reaction, we know that enzymes, if they have to exist and perform an activity, they need a substrate to act upon. So enzyme plus substrate will give us enzyme substrate, and then finally we get enzyme product, and then we get product. Correct, right? So this is how the chemical reaction happens with respect to enzymes. Now, what becomes a limiting factor for this enzymatic activity? Now, the limiting factor means what can avoid or what determines the rate of reaction, or what is the level of reaction it can go. For example, if I have 100 enzymes, and then if I have 1,000 molecules of substrate, now all the enzymes have occupied all the substrate. Correct, right? Can we have any more products? Let's say that 100 molecules of an enzyme in 1 minute can produce 100 products. Correct, right? So now what will happen until we have the enzyme? All these 100 enzymes are occupied in their business with their substrates. So when they go and interact with the substrate, now what will happen? Only 100 we have. That is why there will be no, for example, in 1 minute you cannot expect 200 molecules of a product, right? It's not possible. So that is why the amount of enzyme and the substrate concentration is very, very important. See, when we talk about substrate concentration, the substrate concentration, see, for example, if I have an increase in the substrate concentration, now what will happen? The velocity of the enzyme also rises. So this is a vice versa situation. So when we talk about oh, sorry. Yeah. When we talk about the substrate concentration as well as how we discussed about the enzymatic concentration, so this is how it happens. All the enzymes, let's say that we'll reverse the situation where let's say that I have around 50 substrate and I have around 100 molecules of enzyme. So now what will happen? Only 50 of the enzymes can go and act on 50 on the substrate. Rest, even though we have extra enzyme, we don't get the extra product. So that is how the substrate becomes a very limiting factor, understood, for the enzymatic reaction. Now, another factor with respect to enzyme is the affinity. Okay, for every enzyme, there is a substrate, and it depends on what enzyme. I mean, like for example, if you have a substrate and if you have an enzyme, so this enzyme have to recognize this substrate and act on the substrate as fast as possible. So for this to happen, even though we have the proper concentrations, let's say that we have higher the concentration of the substrate and then we have a proper concentration of an enzyme. Now what will happen? The rate of reaction is obviously higher. More of the products will be produced. Now for this to happen, what should happen? Enzyme should go and react with the substrate. So even this has to become faster. So that property of an enzyme going and recognizing a substrate and reacting as fast as possible is called as affinity. So they go and attract to the substrate as fast as possible. There are many factors which affect the affinity of the enzyme substrate interactions. For example, inhibitors are some of the examples. For like before the enzyme goes and occupies on the substrate, okay, now what will happen? An inhibitor molecule comes and sits there blocking this site of enzyme substrate interaction. So similarly, so two factors, one is substrate concentration, other one is the affinity towards the substrate. Substrate concentration, what happens? The concentration of the substrate becomes a limiting factor. So here we should have more the substrate, more is the velocity of the enzymatic activity. So understood. So that is called as Vmax. So next one, when we talk about the affinity towards a substrate, now what did I tell you? You should have higher affinity towards a substrate for the higher enzymatic activity. So now let's talk about another factor. Another factor I told you what determines the substrate as well as enzymatic interaction is this presence of an inhibitor. What is an inhibitor? See, now we have an enzyme, okay, and enzyme goes and attaches to the substrate. So usually this is the occupying position of the enzyme, I mean like the substrate to an enzyme. Here, similarly, if you look at the inhibitor also have a similar kind of a structure. Now what happens before the enzymatic activity happens and goes and attaches to the substrate? Now what will happen? An inhibitor goes and occupies the position, thereby blocking this enzyme to go and sit on the substrate and cause a chemical reaction. Understood? So this does not happen. So that is how an inhibitor shuts off this enzymatic activity. Understood? So this process is called as inhibition. Okay, for example, we have a very good example, one of the competitive inhibitors. So what is competitive inhibitor? See here, if you talk about a normal inhibitor will go and bind to a different spot, thereby changing the configuration of your substrate, and so that your enzyme cannot go and bind here. Understood? So this is what happens to the inhibitor. When it comes to competitive inhibitors, what they do is they fight for the substrate spot. See here, the structure of this inhibitor and the structure of this substrate is very similar. So this shape similarities, what it does is it will go and occupy the position of the substrate before the substrate comes here. So that is called as competitive inhibition. So one factor is the presence of an inhibitor. So we have two types of inhibitor. We have non-competitive inhibition and we have competitive inhibition. In non-competitive, what happens? We have an enzyme and we have a substrate. So you can see the shape of the substrate and you can see the shape of the enzyme, right? And you see the shape of the inhibitor. Now what happens? Inhibitor goes and occupies this position in the enzyme, that will change its shape or configuration. So when the enzyme and inhibitor combines, the enzyme is no longer having the same shape as before. It will change its structural configuration. So that will make the substrate not recognize this spot, and that is how the enzymatic activity is stopped in non-competitive inhibition. In competitive inhibition, what happens? The inhibitor has a very similar shape to that of the substrate. So what happens? It competes for the spot of the same active site of the substrate. So it comes and occupies before the substrate, thereby stopping the enzymatic activity. So this is about the inhibitor affecting the enzymatic activity. Understood? So a very good example of competitive inhibition, as I was saying, is the malonate. Malonate is a competitive inhibitor. And then for what it is? For a succinate. Succinate is a substrate. Understood? Now what happens here? We have succinate. Succinate, this is a normal chemical reaction or enzymatic reaction. We have succinate as a substrate. We have an enzyme called as succinate dehydrogenase. So succinate dehydrogenase acts on succinate to give us fumarate, which is a product. This is a substrate. This is an enzyme, that is the product. Understood? Now what happens? There is malonate. Malonate is a competitive inhibitor. Now what it does is, malonate goes and occupies the enzymatic positions on the succinyl dehydrogenase, thereby blocking the succinate coming and occupying the position on succinate dehydrogenase. So that will lead to stopping this chemical reaction. Understood? So this is called as competitive inhibition. So did you guys understand this? Yes. Okay. If you have doubts, let me know. Okay. So now we have to talk about classification of enzymes. What are the different types of enzymes we have, or is it only some one type of an enzyme? If we look at all the biochemical reactions, we can classify biochemical reactions based on the type of chemical reaction it is happening. For example, oxidoreduction reaction, or transferase reaction, or hydrolysis, or lyation, or isomerization, or ligation. So all these are chemical reactions. Based on what type of chemical reactions or biochemical reactions they can conduct, or based on its activity, enzymes are classified into these types. What are those types? One is oxidoreductase or dehydrogenase, transferase, hydrolases, lyases, isomerases, and ligases. So these are the different types of enzymes. So based on its activity only, or based on, for example, if you talk about the prior example, we have succinate. Succinate is a substrate. So it goes and binds to the substrate. That's why the enzymatic name comes succinic. Okay. Next one is dehydrogenase. Dehydrogenase is a type of chemical reaction this enzyme takes place. And by the end, we give ACE as the nomenclature, common term for all the enzymes. So the first word of the enzymes, okay, is nomenclated based on the substrate it acts on. So the second word of the enzymes is given based on the chemical reaction it conducts. Okay, this is the nomenclature of enzymes. So let us see how each of these enzymes in this category conduct a chemical reaction. For example, first one we have oxidoreductase or dehydrogenase. Okay, so what it does is it catalyzes oxidoreduction between two substrates. For example, we have, for any chemical reactions, we have two substrates, isn't it? So we have 1 and 2. Okay, one of the substrate is reduced, and the other substrate is oxidized. So if I have A plus B is equals to A plus B, let's say that. First of all, A is reduced and B is oxidized, and then later A is oxidized and B is reduced. So this is called as oxido reduction reaction, and these enzymes are called as oxidoreductase or dehydrogenases. Understood? So next one is transferases. So what does it do? It transfers as one group, okay, a prosthetic group or any group, it transfers from one substrate to another substrate. For example, if your substrate had a group attached such as G, and then it reacts with another substrate S. So now by the end of the reaction, what happens is you have the transfer of the G group to this S, and the normal S comes, S and S dash, you can see here, right? So that is called as transferase. A group is transferred between one substrate to another substrate. Next one is hydrolases. So what does it do? It adds water molecule. For example, we have peptide bond, or we have glycosidic linkage, or carbon-carbon bond, or carbon halide bond. Okay, so all these bonds are there. So what happens? Let's say that any of these bonds would be in this nature, correct? Right? Peptide bonds or glycosidic bond. So here what happens is a water molecule is added. When it is hydrolyzed, when the bonds are hydrolyzed, the bonds are broken. So that gives, okay, AOH, and then we have our liberation of one hydrogen molecule. Okay, so this is called as hydrolases. Next one is lyases. So what happens in lyases? See here, if we have our substrate attached to a particular group, now we have X and Y, and we have our C and C in between. Now lyases, what it will do is a group is removed. Okay, so they perform the mechanical reaction where a group is removed by hydrolysis. Okay, see here, the carbon-carbon is removed, and then it leaves a double bond. So this is called as lyases. Understood? Next one is isomerases. So what is isomerism? So we have a physical property which is a geometrical isomerism. A molecule shows a specific kind of a rotation, which is like the position of these groups can be in the same side or in the different side in the branch structure. So based on that, we have cis isomerism and trans isomerism, and then we have these isomerism. So if any of these isomers are able to change the isomerism, geometrical isomerism, for example, cis to trans or trans to cis, so these are called as isomerase. Okay, see here ABC, and then it changes to the different orientation, that is BCA. Understood? So this is called as isomerases. Next one is ligases. So ligases are also called as gums. What it does is it glues two different groups together or two compounds together. For example, if you have X plus Y plus ATP, now what it gives is XY plus ADP plus phosphate. So this is called as ligases. Okay, chalo. So another important factor, if you observe the characteristics of an enzyme, many of the enzymes are produced in the form of apoenzymes. Understood? Before we go into this, are you guys clear with the prior topic? Okay, good. Thank you for the response. Okay. Yeah. Another important feature of the enzymes is, if you look at any biological systems, okay, an organism, what you see is many of the enzymes are not produced as active enzymes, they are produced as apoenzymes. So what are apoenzymes? Apoenzymes are the protein component of the enzyme, which is in its inactive form. To make this apoenzyme active, a prosthetic group or a cofactor comes and attaches to this to make it an active enzyme. So these are called as cofactors. Making an apoenzyme a holoenzyme, okay, is called as a cofactor. Understood? So what are cofactors? I told you that apoenzymes are the protein part. Cofactors are the non-protein constituents of the enzyme. So what happens with respect to why we need cofactors? So now the cofactors just attach to this apoenzyme, and then they make it as holoenzyme. Understood? Or active enzyme. Now, based on the chemical composition, we have different types of cofactors. So first one is prosthetic group, second one is coenzymes, and then we have metal ions. So let's see what are those. First one is prosthetic group. What is a prosthetic group? They are organic and they're tightly bound to your proteins. Okay, So that is to your apo enzyme. For example, heme in your peroxidase and catalases. So this is an example. Heme. This is a heme. So this heme group in peroxidase and catalase, what they do is they help breaking down this hydrogen peroxide to water and oxygen. Okay. So this is also called as catalytic activity. Understood. So that is what is prosthetic group. Next one is coenzymes. When you listen to the term called as coenzymes, you have to think that okay, your memory should go to this word called as vitamins. What are coenzymes? Coenzymes are nothing but vitamins. Now you understand why we have to take protein, vitamins a lot, on a regular basis, because many of your biochemical reactions are dependent on vitamins. You have so many vitamins: A, vitamin C, vitamin B, vitamin D. You might have come across all these names, right? So what are those? Those are called as coenzymes. So whenever we have an organic group, and they're called as transient binding. What is the meaning of transient binding? Now, if you have an apoenzyme, so to make this a holoenzyme, these cofactors are produced only when it is required for that particular chemical reaction. Understood? That binding happens only when that particular enzymatic activity is happening. So that is called as transient binding. Understood? So it transiently binds to this apoenzyme, so which is an organic group, that is called as a vitamin, okay, which is a coenzyme. Now, for example, you have nicotinamide, adenine dinucleotide, NAD, and then you have NADP. So both of these enzymes, they contain niacin as the cofactor. Understood? So this is called as cofactors, which are vitamins. Next one is metal ions. So let us take an example of one more enzyme, that is carboxypeptidase. The role of carboxypeptidase is very simple. We have amino acids. Amino acids has N terminal. Sorry, amino acids have N terminal and they have C terminal, right? So in the C terminal, which is also called as carboxy terminal. So here what happens? A peptide bond is formed between another nitrogen group, right, or amino group. So here it goes and breaks this bond. So that is called as carboxypeptidases. Understood? So if you talk about this enzyme, you can find that zinc is a cofactor. A metal ion binds to your apoenzyme, okay, to convert this apoenzyme into holoenzyme. Understood? So this is called as, so these are metal ions. Okay. So here we come to the end of biomolecules. Okay. When we have time, we will just revise it as a crash course when to boost up our memory and our concepts. Okay. So if you have any doubts, please ask me before we go into the next topic. Can we go to the next topic? Students, can you hear me? Are you there? Okay. So you don't have any doubts. You can just put the reactions. You have the emojis. You can do like this. Okay. It's simpler and it's faster. All right. So let's go to our next topic. We'll have an introduction, which is a little bit interesting. Not a little bit, very interesting. Okay. So we'll talk about one of the most exciting topics, which is the cell. Okay. You might be confused why we started biomolecules and then we go for cell, the unit of life. The reason is very simple. If I directly start with cell cycle and cell division, that will be very difficult for you to understand. So let us have a very, a beautiful understanding about cell first, and then we will try to understand how cell division occurs, right? So first, let's talk about our topic, that is cell structure and function. So why do we have to study cells? Okay, we'll talk about this. Why do we have to study about cells? Now, if you look at all these organisms, and if you look at your non-living organisms, what is one thing that you differentiate? You differentiate that even though you have many of the functions that are, for example, growth, reproduction, response to stimuli, homeostasis, biochemical reactions. You might give so many examples to tell the differences between the living and a non-living organism. Now, one of the major fundamental difference that you will also come across is the presence of a cell in a living system and an absence of a cell in a non-living organism, non-living things, right? Or abiotic things. Now, what is a cell and why do we have to study? See, when you see the entire biodiversity that is present around us, we have from unicellular organisms to complex eukaryotic multicellular organisms. Now the two questions arises: how a small unit is able to perform a gross total of all physiological reactions, such as it does reproduction, it prepares food, does transportation, it responds, it can think, it can communicate, right? It does so many different functions. So to accomplish all these functions, so it has to perform some physiology. So a study of a cell is more about a physiological aspect of how the cell behaves and how the cell is able to perform all this activity to accomplish all this that we see in our living world. Now, another important question that curates our mind is what was the necessity or why is it so important for this cell to establish, okay, a multicellular structure? Why did a cell decide to form tissues, to organs, to organ systems, to organisms? Or it became more and more complex. And as it became more and more complex, cell differentiated and their responsibilities changed. So even though all cells perform all the activities as a unicellular organism, or a unicellular organism performs all the living activities like you do, okay, so vice versa. So if you observe the the cellular activities, what you understand is you have a large amount of diversity in their chemical composition, and that has influenced its activity, and that is how the cell differentiated in its function, its shape, structure. Everything varies according to its location, its function, understood, and which organism it is present and its complexity. Correct, right? So it's a very wonderful structure to study in all of biology, because this is where all the life started, and this is how the life has complexed, and that is what has resulted to entire thing that we see today. Okay, so to define cell, it's very easy right now. Cell is a structural and functional unit of life. Okay, so we've been practicing this definition for a very long period of time, right? So if you just take a pause and then look back, how did we come to this statement of cell is fundamental structural and functional unit of all living organisms? So there is a history. So we did not just see cells through our naked eyes and then we just named it, okay, this is a cell and this is how the cell structures are. It is evolution of science as well as evolution of technology that has resulted in more understanding, okay, about cells. All right. So now we have more understanding about cells as the time passed because of the advancement of technology. But we cannot forget what we have been through from some years, right? So we have evolved. So if you go back to the history, the first person we have to thank about is Anton von Leeuwenhoek. So Anton von Leeuwenhoek was a curious biologist and is a lens grinder. Okay, so what is a lens grinder? You might have seen the magnification glasses or the people who wear the optics. So he is a very exceptional lens grinder. So his hobby was to create lenses that are more and more powerful in magnifying capacity, and eventually he ends up making a first very simple microscope. Okay, so with this discovery, Anton von Leeuwenhoek became very curious. Now what he did was he started examining his environment, which is very common to him, like wherever he lives. For example, he was examining the water from the well, he's examining the fecal matter, the scrapes from his teeth, the blood cells, okay, the fluid of insects. Okay, so whatever he can get his hands on, he wanted to see what's under this lens and he wanted to see if there is anything present. To his surprise, he observed a lot an entirely new different world under the microscope. For the first time, a human being is seeing something which he cannot see through his naked eyes. So that's what opened the door for this field of microbiology and cytology, and what understanding we have about life also. Okay, so Anton von Leeuwenhoek's observation includes many of the bacteria, the teeth plague, the blood cells, red blood cells, etc. But he was not able to, give it a name, and he understood this. Okay, these are the microbodies that are invisible to our naked eyes. And inspired by the Anton von Leeuwenhoek work, now the Robert Hooke also, what he does is he develops more powerful microscope, and he would be examining a lot of plant material, and he stumbles upon these cork cells. Okay, so when he takes the cork cells and he observe it under the microscope, what he sees is it's like a honeycomb. So you can see that it's like a honeycomb structure, right? So with a very repetitive structures that are repeating. So when he observed this structure, what he thought was, okay, they are like small units that have been repeated, and he called this small units as cells. Okay, he named it as cells. So after the work of Robert Hooke, okay, and he discovered cell, now a lot of curiosity that arose in the scientific community. Many of the botanists, see, we had biology, and biology was split into two major groups. One is botany, other one was zoology, and all of them were excited all of a sudden to see whether it is an animal, whether it is a plant, either they were wanted to study the characteristics of cells. Okay, like it started from microorganisms only, and even the higher organisms. So during this time in 1838, okay, during this time in 1838, we have two important figures. One is Mathis Sheldon. So Mathis Sheldon, okay, so this is M.J. Sheldon or Mathis Sheldon. Mathis Sheldon is, he's from Germany, okay, he's from Germany, and he was a botanist, and he started observing a lot of plant material. So he's examining a lot of plant material, and what he observed was in different samples on in different specimens that he was taking, all of them were composed of cells. So with this, he comes up with a hypothesis. Okay, all the living constituents, whether it is animals or plants, all of them have to be composed of cells or the products of cells. Okay, so that is what the statement our Sheldon will come across. Now comes Theodore Schwann. Theodore Schwann is from British. Okay, he's a British zoologist. So he and Sheldon actually shared this idea about their observations. He observed animal cells and he observed plant cells, and they came up to this conclusion that there is a significant difference between the animal cells and the plant cells. Even he agreed, according to his animal cells observation, that all the cells in the animals are made up of cells or the product of cells. He says all the plant cells, okay, is either made up of cells or the product of cells. Okay, so both of them combine and propose this hypothesis. So after proposing this hypothesis, one thing they could not explain us is how was the first cell formed? How did the cell even become existing? Okay, if there is a cell, it has to come from some place. How did it come? So they did not explain that. Understood? So this led to the way to a very famous theory that is called as cell theory. Okay, so later what happens is people were wondering, okay, all these living organisms that we see is just so complex. All of us are made up of, excuse me, yeah, sorry students, I thought we ran out of time. Okay, so all of it is made up of cells and its products, and they could not answer how the origin of the cells took place. Now Rudolf Virchow, he comes up in 1855. See, there is a lot of time difference, right? It took almost 20 years to realize that. So he hypothesized that cell division happens and the new cells are formed. So he says that the new cells, the present cells, are the result of pre-existing cells. Okay, so he calls in his language that is omnis cellulae e cellula. That means the cells are formed from pre-existing cells. So the prior cell theory hypothesis was again modified. with the first statement being all living organisms are made up of cells or the product of cells, okay, was further extended to another statement, that is all cells arise from pre-existing cells, to complete the hypothesis of a cell theory, right? So this is what led to the beginning of understanding cells even more and more. Now, when we talk about the cells, we understood that there was a journey, and in this journey we had the contributions of this curious scientist that led us to the discovery of cells, and we also understood some of the characteristic feature of the cells. As we started studying the cells, so did the microscopy also increased. So this microscopy right now is a very important tool for us to understand the structure, the functions, and the characteristics of the cell. Now, just taking the microscope, I will tell you how the technology is right now with respect to microbiology. So here, first we had simple microscopes or hand microscopes. The hand microscopes developed into compound microscopes that we use in our laboratory. So each of the microscopes have magnifying capacities of either it is 100x. Let's see, for example, if you tell 10x, okay, 1 second. If you have 10x as the that is present in your eyepiece of your microscope, so then you put the object lens that is also 10x. So finally you will get 100x. So what does it mean? If you see in the eyepiece and if you see in the object lens, now what will happen here? It will be written 10x, and in the object lens also it will be 10x. That means it can magnify 10 times. 10 into 10, 100. So you will see the image 100 times. Similarly, if I talk about 45x, and now I have a 10x eyepiece and then I have a 45 object, so that will give me 450 times the magnification. 100x is 1000 times. So similarly, the compound microscope also became more and more powerful. Now, with the compound microscopy being developed, now we went on to further technological advancement. One of the greatest one is electronic microscopy. So in electron microscopy, what we do is the electrons are bombarded in a vacuum at a high velocity onto the cell. Now what happens? The electrons will penetrate or bounce back. Based on that, we establish a computational image. So here, the size of electrons are very, very negligible. So because of that, we get a very detailed image of the cell, okay, using computer as well as the electrons in microscopic technique. Now we have fluorescent microscopy also, where we can dye a specific cell organelle, okay, or a specific part of the cell. We can observe cell division by using fluorescent dyes, right? Dye coloration, so that can go and bind to some of the cellular organelles, and you can observe its cell division, okay, and or the movement of certain biomolecules, etc., in our fluorescent microscopy. Understood? So this is a small trend, not small trend, a major technological leap that has taken in the field of microscopy. Now, why did we talk about it? Now, with the advancement of microscopic technology, where it is pure physics and biophysics, and then you have the biochemistry applications also, you have to understand the characteristics of cell to develop a strain, okay, to label a specific part, etc., right? So that's what happened with respect to the establishment of this microbiology field, and then that led to the characteristics and specificities of the cell, which we'll discuss in the coming class. Okay, we'll take our time and understand the structure of cell and differentiate into prokaryotic and eukaryotic cells and study their characteristics. Okay, students, so if you have any doubts about today's class, you can ask me. And some of you had asked me about the plant kingdom, right? I'm sorry, I just forgot about it, and I will take this in tomorrow's class, or when the time permits. Okay, the life cycles of plants, I remember, I'll get it done. All right, students, thank you. So if you have anything doubts, you please let me know. Okay.